consider the following.
Today we're going to talk about iron oxide nanoparticles. So what are iron oxide nanoparticles? Well, first of all, nanoparticles, they're any particle or object that's between 1 and 100 nanometers in size. What we did in this experiment is we made nanoparticles, and they have magnetic properties, and we put them through an NMR test. NMR stands for nuclear magnetic resonance. So what that means is that the nuclei of an object, let's say either nuclei right here, they travel around in a circle or any direction like this, either way, and they have enough energy to create electromagnetic radiation. This radiation can be used for these purposes in the real world: human tissue analysis, or eating blood vessels, or even just nanotechnology like semiconductors.
The variation for the 0.8 ratio was that we added 5 milliliters of 2 molars of iron chloride to the jar instead of 2.5 milliliters, and the synthesis was then adding the normal 25 milliliters of ammonia, NH3, and that was when the nanoparticles started forming. After creating the nanoparticles, we could see they had magnetic properties. When taking the magnet to the jar, we could move the nanoparticles around the jar wherever we made the magnet move. This was also true for the bio students who performed testing on the dilution with the shrimp when a magnet was introduced into the experimental shrimp's environment. Then the shrimp would also follow the magnet.
The effect of doubling the 0.4 concentration of the nanoparticles to 0.8 will double the magnetic strength. The lethal dosage calculated by the biology group revealed that 0.8 variation in the nanoparticle killed 100% of the brine shrimp population, with only 1 milliliter of nanoparticle mixed with 1 milliliter of seawater. The best contrast agent would be the 0.4 concentration of the nanoparticle, because it requires the least dilution of the solution for it to be considered safe. The most toxic variation will be the 0.8 solution, because it needs to be the most diluted for it to be considered safe.
Brine shrimp make great model organisms as they can tolerate levels of salinity ranging from 25% to 250%. Millions of brine shrimps can be reproduced very easily, which allows for testing many environmental pollutants under controlled conditions quickly. They can also adapt to levels of salinity very quickly in order to better produce their chances of survival and reproduction. Their living conditions are simply saltwater, so they can be kept virtually anywhere, and you can keep very many of them in a small space, as brine shrimp are very small in size. As chemistry mentioned, the concentration of the solution is 20 milligrams per milliliter. Due to the different concentrations and level of dilution in each sample, hypothesis is that the toxicity of the solution would decrease according to the amount of dilution associated. This would mean that the 1-to-1 solution would have the lowest percentage of shrimp alive, and 1 to 1,000 would have the largest percent of shrimp alive. The design included 5 test tubes from concentrations of 0, 1-to-1, 1-to-10, 1-to-100, and 1-to-1,000. The first sample included 2 milliliters of seawater, while the second sample had 1 milliliter of seawater and 1 milliliter of the iron oxide solution. Every successive tube after that has 9 milliliters of seawater and transfers 1 milliliter from the previous tube. So for example, the 1-to-10 solution, 1 milliliter was used into the 1-to-100 test tube. This would mean that the concentration would decrease from a magnitude of 10 from the 1-to-1 solution all the way down to the 1-to-1,000 solution. So the 1-to-1 solution would have a concentration of 20 milligrams per milliliter, and the 1-to-1,000 solution has a concentration of 0.02 milligrams per milliliter. Each tube was then placed into a well with the corresponding concentration. All the wells included 10 brine shrimp present at the beginning. The shrimp were then observed over the next 3 days, and the percentage alive was recorded for each day.
After 72 hours, it was observed that the 1-to-1 dilution population was the most affected by the high dose of nanoparticles, killing 90%. The shrimp in this solution were mostly covered in the black solution and started to lose motor skills. As the nanoparticle sample was decreased in the dilution, the more likely the shrimp were not affected. For example, the shrimp in the 1:100 and 1:1000 moved faster and survived longer than the 1:1 dilution and the 1:10 dilution. After observing the brine shrimp in the experimental environments, the data concluded that the 1:1000 dilution of the nanoparticle sample would be the lethal dosage in order to kill 50% of the population. Since the 1:1000 killed 50% of the population, it would be considered the lethal dosage of 50% of iron oxide nanoparticles, because any dose lower would harm less than 50% of the population. Based on the following trend, the best nanoparticle would require the lowest toxicity, 0.02 milligrams per milliliter, opposed to the 20 milligrams per milliliter. Since 0.02 concentration of the 1:1000 killed 50% of the population, is considered safe. Also, basing this experimental data off the brine shrimp, it would be acceptable to introduce this to the shrimp population. However, in other organisms, a larger concentration would be required for a significant effect to be obtained. For example, the lab rat is required a 6,200 milligrams per milliliter for the lethal dosage of 50% of the population, opposed to the 0.02 milligrams per milliliter in the shrimp.
We can use what we have learned in brine shrimp nanoparticle testing in medical applications on humans. Learning the LC50 and LD50, we will be able to figure out the effects on where 50% of the subjects die, therefore knowing the safe dose. Nanoparticles can also have positive effects in contrast agents and imaging. Drug and gene delivery into tumors is perhaps its most ideal effect, as it can be used to save millions of lives and further advance medical research.